Hey everybody, so I wanted to talk a little about the development of nervous system, specifically how our nervous system gets patterned. We are going to go through some interesting concepts here. Let's start with the three layers of cell that a vertebrate embryo has. That is ectoderm, endoderm and mesoderm. Now the nervous system develops from the ectoderm. That is a narrow central slip of ectoderm flattens out to become the neural plate, which then invaginates, forming the neural groove, and finally, through the process of neurulation, a neural tube is formed. Today, we will largely talk about the first step of this process, that is, the formation of the neural plate, and what is meant by the organizer region. For that, let's head back to 1924 when Hans Spearman and Hilde Mangold conducted a groundbreaking experiment. The experiment involved transplanting small pieces of tissue from one amphibian embryo to another. One aspect involved transplanting the dorsal lip of blastopore, which forms the dorsal mesoderm, from the pigmented donor embryo to the ventral side of an unpigmented host embryo. This dorsal mesoderm underlies the dorsal ectoderm, which gives rise to dorsal epidermis, including the neural. After transplantation, they found that in the unpigmented host embryo, ectodermal cells were induced to form a virtual copy of the nervous system, and thus they named this donor tissue the organizer. They later proved that the dorsal lip of blastopore was the only tissue having this organizing effect. This study demonstrated how induction, a process by which cells of one tissue direct the development of neighboring tissues when the two comes into proximity, is very important for the development of nervous system. So from this experiment, we realized that how important the process of induction was to the formation of neural plate. But we were able to delineate further details about the patterning of nervous system only due to the advent of molecular biology and better availability of markers of early neural tissues. When an ectodermal cell was isolated and thus preventing cell-to-cell signaling, it was found that the cells readily acquired neural properties. This meant that the default fate of these cells was in fact neural differentiation. Therefore, the inducer that we were searching for was actually the de-repressor. The next logical step was to find out the molecules responsible for repressing the neural differentiation. Here, let me introduce BMPs, or bone morphogenic proteins, which are members of the TGF-beta related proteins who act via the serine threonine kinase class receptors. It was found that in the absence of signals from organizers, ectodermal cells secreted BMPs which suppress neural differentiation. This finding was confirmed when a truncated version of BMP blocking its signaling triggered differentiation of neural tissue in Xenopus frog embryo. The question that follows is, how does organizer de-repress? Organizers secrete many proteins such as follistatin, nogin, cordon, which antagonizes the BMP signaling. And here it was also found that exposure to FGFs is also a necessary step in neural differentiation. So we have reached this step where we have formed the neural plate, but then how does the variety of cells from the rostral region to the caudal region develop over time? This is something that we will see in the next video. The reference used for the video is Principles of Neural Science, the 6th edition. I hope you guys like the video. Thank you so much.